will discuss about eukaryotic cell structure with respect to cell surface structure and their variations. We all know that the eukaryotic cells were evolved at around 2 billion years ago. Some of the organelles of the eukaryotic organism were evolved by endomembrane system and some of the organisms were evolved via endosymbiosis. First, eukaryotic cells were unicellular, but when they colonize together, they give rise to the idea of multicellular eukaryotic organism. Today, we will discuss about the cell surface. That includes cell wall, cell membrane, the junctions among different cells as well as cell surface projections. When we are talking about cell wall, we will talk about fungal cell wall, algal cell wall, plant cell wall as well as their variations. It is very important to say that the animal cell and protozoa cell lacks any sort of cell wall. We will also talk about different protist cell wall. Fungal cell wall structure. So, when we are talking about fungal cell wall, so we have to remember about two main components, glucan as well as chitin. There are variations among fungal cell wall. In case of true fungi, we can find the presence of zymogen. In case of slime mold, we can find the presence of cellulose, and in case of fungus like protist, we can find cellulose as well as chitin. Now, this chitin and cellulose make polymeric fiber and they are attached to the amorphous glucan. In case of the hyphal tip, we can find the two layers. The outermost layers are having much more glycoprotein than that of the inner side. So, in case of fungal cell wall, we can find the presence of glycoprotein as well along with the different kinds of polysaccharide, chitin and glucan. Fungal cell wall function. So, with such composition and alignment of the composition, the fungal cell wall renders some of the functions. It is the attachment site for some of the enzymes. It interacts with the neighboring cell. It protects the cell. It gives the mechanical barrier and it actually gives the shape to the cell. Now, when we are talking about the algal cell wall, the volvocells have proper cell wall, but in case of some of the Chlamydomonas species, we can find the cell wall is not made by the continuous polymer. There are some of the proteins like extensins, they make the polygonal structure and they are arranged in the plates. We can find the presence of sporopollenin like isoprenoid chains over there. When we are talking about the composition of the algal cell wall, it consists of several structures like mannan, we can find xylene, alginic acid as well as we can find the presence of sulfonated polysaccharides. Now, when we are talking about mannan, it is made by the mannose residues. So, the manosyl residues are arranged together. Now, when we are talking about the xylene, they are actually the polymer of xylose residue found in case of red algae as well as in case of brown algae. We can find the presence of alginic acid. So, alginic acid is polymer of D-glucouronic acid, galactouronic acid as well as mannuronic acids. Now, apart from alginic acid which is found in case of red and brown algae, we can find the presence of sulfonated polysaccharides. Now, agarose is the example of sulfonated polysaccharides. It is found in case of some marine algae, Giladium corneum. Carrageenan is another sulfated polysaccharides. Apart from the polysaccharides and glycoprotein, we can find the presence of calcium as well as silicon. Hydrodiction cell wall is made by silicon, and we will talk about diatom later, which is also made by the orthosilicic acid. Now, apart from this, I have already told you sporopollenin is present, which is a kind of isoprenoid chains. And apart from all these general structure, we can find the presence of calcium also along with silicon. So, calcium is present as calcite or argonide in case of some of the chlorella species. So, algal cell wall function. Again, this algal cell wall is important for cell to cell communication as well as receptor mediated some of the transport, and it is again the binding site for some of the enzymes as well. Next, plant cell wall. Now, when we are talking about plant cell wall, let's talk about its model as well as biosynthesis. So, plant cell wall is made by mainly three types of layer. In between two junction, there is a amorphous thin layer called middle lamellae, which is actually first made, over which the primary cell wall is getting deposited, and ultimately when the primary cell wall is getting completed, then only the secondary cell wall is made. Now, when we are talking about the model, first we will talk about the overlayer of this middle lamella, then over which primary cell wall is getting deposited and expanded. When we are talking about the biosynthesis, the cellulose are deposited over the plasma membrane, right, and they will make the rosette-like structure. Then through ear as well as Golgi by vesicles, when the glycoproteins are getting inserted within the cellulose structure, they will give rise to the structure of plant cell wall. Not only this, but also in case of plant cell wall, we can see the destruction of the older one can give rise to the new cell wall by the acid hypothesis. So, H plus ATPase or proton ATPase along with the expansion protein is actually mediate this kind of model for the plant cell wall biosynthesis, and by the overlayer of different layers actually the plant cell wall is made. Primary cell wall. So, primary cell wall is actually found in case of growing cells, meristematic tissue, succulent tissue, etc. And it is mainly made by the pectin, hemicellulose as well as cellulose. Now, cellulose is very important beta 1,4 glycosidic linkage containing polymer, polysaccharide rather. The cellulose that is fundamentally found in case of plant cell wall, it gives the strength to the plant cell wall, and primary cell wall is first made, it is overlaid, and when the thing is getting completed, then only the secondary cell wall is getting formed. There are two types of primary cell wall. In case of general flower plant, we can find the primary cell wall 1 type, and which is made by pectin, cellulose and hemicellulose, but in case of primary cell wall 2, which is actually found in case of grass family and poaceae, we can find again the presence of pectin, hemicellulose as well as cellulose, but their components, their concentration, their percentage varies. Secondary cell wall and middle lamella. So, I have told you these during cytokinesis as plate, first the middle lamella is overlaid over which the plant primary cell wall was made. So, we can find this kind of thin amorphous middle lamella in between two plant cells. They are made by the pectic acid or calcium magnesium pectate as well as pectin. They are some amorphous thin layer. But the secondary cell wall is made after completion of primary cell wall synthesis, and it is made mainly by lignin, which is the phenol derivative component, which actually helps the inhibition of water evaporation from the plant cell. Function of plant cell wall. So, the plant cell wall actually helps to maintain the turgor pressure of the cell. Not only that, it gives the strength, it maintains the shape, plant pathogen interaction is mediated by plant cell wall, cell signaling happens, the transport occurs, cell to cell communication happens, as well as plant cell wall in the lignified suberinized cell wall, it inhibits the evaporation of the water. But there are variation of eukaryotic cell wall. In case of Euglena, the flagellated protist, we can find the pellicle-like soft cell wall structure, not proper cell wall. So, it renders the flexibility and contractility to the cell. Diatom, which was first thought inside the algae family, now it is the separate protist. It contains orthosilicic acid over its membrane, and you know that when it is getting synthesized, it is less energy consuming. It promotes its rapid growth rather. In case of dinoflagellate, we can find the plate-like cellulose made structure over their cell wall, and we can follow the theca structure over there. So, they are all the variations of eukaryotic cell wall. Eukaryotic cell membrane. Now, first let's talk about its model. In 1925, Dabson and Danieli proposed the rigid model of cell membrane. But then the fluid mosaic model at around 1972 was hypothesized and confirmed by Sanger and Nicholson. We know the components of the plasma membrane includes lipid, protein as well as carbohydrate. So, lipid makes the sea over which the proteins are floated. The lipids may be phospholipid, sphingolipid or maybe sterols. The sterol along with the unsaturated fatty acids, they keeps the fluidity of the membrane. As we know, rigidity as well as fluidity both are needed for the many of the physiological functions of the plasma membrane, and they are actually maintained by unsaturated fatty acids as well as cholesterol in case of animal cell. You know, the cell membrane is ubiquitous. It is present in animal cell, protozoa cell, protist, algae, fungi as well as plant. So, it is present everywhere. Now, apart from these three types of lipids, when we are talking about the plasma membrane protein, they either may be extrinsic, associated by loose polar interactions. They can be easily removed by sodium chloride extraction, or may be intrinsic, associated via non-covalent or covalent interaction. The transmembrane protein travels throughout the membrane. They are engaging to make the non-covalent type of interactions with the fatty acyl chain, but integral monotopic protein, they are bound by the covalent interaction of any one of the leaflet. Apart from lipid and protein, which varies according to the type of cell, we can find the presence of carbohydrate, which makes the antigenicity of the cell or marker to the cell. It helps in the cell-to-cell recognition process. It acts as receptor to bind with the ligand. So, we all know about the blood group antigen, which actually is served by the carbohydrate. If the carbohydrate is attached to the lipid, that is called glycolipid. If it is attached to the protein, that is called glycoprotein. Function of cell membrane. So, what I have told you, the transmembrane protein which is attached to the lipid membrane via non-covalent interaction. So, it makes the channel protein as well as carrier protein for the passage of ions as well as macromolecules actively or passively. Apart from the transport, there are so many other functions of the plasma membrane or cell membrane rather. It acts as the platform for the propagation of signal transduction, nerve impulse transduction, etc. Plasma membrane also maintains the osmolarity and it actually separates it from the outside environment. There are variations of the plasma membrane structure because according to the need of the cell, whether it is giving the insulation, whether the enzymatic properties are needed for the physiological purposes, lipid to protein ratio or concentration varies from 4:1 to 1:4. Over the myocytes, the sarcolemma is found. Over oocytes, ulema is found, and over the axons, axolemma is found. It is very interesting to know that historically the plasma membrane was known as plasma lemma. Junctions are the adherent sites in between the cells. So, we can find different types of junctions among the eukaryotic cells made by different type of surface protein. The first, tight junction. In case of tight junction, we can follow where the epithelial lining of or the sheaths of cells are there. These type of junction makes the tight contact in between the epithelial cells, and it makes the lumen facing site is called apical site and the external facing site is called basolateral site. Now, this tight junction is pivoted by many of the interlocking junctional proteins, and they inhibit the movement of transmembrane protein according to its function as well as the passage of macromolecules to the two adjacent neighboring cells. And we know that in case of lumen facing site, the transmembrane proteins are engaged in case of receptor mediated endocytosis, and to the basolateral sites the proteins are engaged to the exocytosis. So, by layering of such tight junction, two functions can be separated easily. The gap junction. Gap junction is what? Gap junction is made by the different types of microtubule proteins. Okay, so these sort of microtubule proteins is actually help to propagate the action potential in case of the cardiac muscular cell. So it helps to propagate the action potential, nerve impulse, as well as cardiac love loop, etc. So what is happening here in case of gap junction? The connection proteins are making this sort of microtubule structure in between two cells, so easily action potential can be propagated through. Adherent junctions. Adherent junctions can again be made in between the cardiac cells, in between the synapses. So what is going on? There are two types of protein. The junctions is made by the cadherin proteins, right? And the catenins proteins are there inside the cell, which anchors the plasma membrane protrusions or the junction made in between two cells. Desmosome. What is desmosome? So desmosomes, again the junction type in between two epithelial cells, and this particular desmosome is made actually by some sort of cadherin proteins again, but it is tethered with inside the cell by keratin like microtubules, and it is actually found in case of skin cells, the epithelial cells present in case of skins. Hemidesmosome, right? Hemidesmosome is made by integrins like protein. So again it is found that epithelial cell are attached to the basal lamina here, and again it is making some sort of junction or some sort of connection in between two adjacent eukaryotic cell for the propagation of macromolecules as well as signal. That is very important for the signal propagation. Now we can follow the proteins which actually make this sort of microtubule for the connection, for making the junctions. If against them the antibodies are raised, so genetical abnormalities can be happened. Plasmodesmata. So that is related to the plant cell. So by desmotubules, this sort of plasmodesmata connection is made, which actually helps the transport of macromolecule, tRNA, etc. in between the adjacent cells. And in case of plant, it actually helps the transport among different neighboring cells. In case of plant, we can find apart from these junctions some sort of cell surface projections. So in case of root cells, we can find the presence of cilia and cuticle. So in xerophytes, to inhibit the evaporation of water, some waxy layer is present. The animal cell surface appendages or protrusions. Now, flagella and cilia are actually togetherly called as cell surface projections. In case of animal cell, they are told together as unduly podia as the cell organelle rather, and it is actually tethered to the another types of cytoskeleton related to the centriole like microtubule. Now we can find in case of flagella, which is 10 times higher than that of the prokaryotic flagella, it helps in case of swimming of spermatozoa cell. You can find it here, and they have 9+2 arrangement. That means 9 pair of microtubule of flagella. It is surrounded 1 pair of microtubule, and for its motility, the dynein protein is there by ATP hydrolysis. It helps during the course of swimming action. Cilia. The number of cilia is much more higher than that of the flagella, and it actually helps in case of filtering, feeding by absorption and in other physiological processes. And cilia also follows these types of 9+2 microtubule arrangement. But we can find various types of cilia as primary cilium over different cell surface. So let's talk about the parallel to that of the cilia. Primary cilium can be found in case of mechanoreceptors over the kidney tubules. In case of microvilli formation, intestine, stomach and kidney tubule over the surface, we can find the presence of such mechanoreceptors, the undulation of which actually helps to propagate the signal. We can find the presence of chemoreceptor over the odor neurons, as well as we can find the presence of photoreceptor over retinal cell. They help to propagate the signal by their undulation. So, it can open some of the sodium potassium channels for the propagation of nerve impulses. So, they are the parallels to that of the cilia. It is very interesting to know that in case of microvilli, over the surface we can find the presence of the glycocalyx like material, right, which actually help them for protection, adherence as well as transport. In case of junction material, we have talked about extension, we have talked about integrins, cadherins, caterin, connections, etc. So, they are the surface glycoproteins which help to make the junction structure for the transport purpose, for communication purpose. There are so many other cell surface marker glycoproteins also which actually help in case of physiological processes. They can be told together as cell adhesion molecules or CAM. Selectins, which are fucosylated glycoproteins, right. It may be E-selectin over the endothelial cell, P-selectin over platelets or L-selectin over leukocytes. So, they help in case of recognition. So, they actually also help during the course of homing of lymphocytes. Immunoglobulins, very important glycoproteins over the immunological cells which go and bind to the antigens for the propagation of immunological responses. Apart from this cell surface projection or protuberances, there are so many differences, not differences, the terms related to the cell surface. So, the invagination of the cell membrane inside is called as caveoli within the cytoplasm. So, they help during the course of endocytosis and exocytosis. Caveoli is made by the caveolin protein and it is rich with sterols, cholesterol mainly, which help during the course of signal transduction. Apart from these, the microtubule made cytoskeleton is there. Cytoskeleton is made by the actin filament and in case of RBC, it is made by the spectrin. It tethers the lipid membrane, protein as well as lipid towards the cellular matrix so that it cannot diffuse throughout the membrane. So, this is called cytoskeleton. It helps to anchor the cell surface material inside the cell. I have told about the eukaryotic cell surface structure including cell wall, cell membrane, junctions, cell surface projections, right. And I have talked about differential cell wall structure in algae, in fungi, in plant, and I have talked about the structure and function of the various cell membrane along with the junctions made among them, between them, their functions and cell surface projections. Thank you.